Hi, everyone. My name is Kara Bourne, and I'm one of the second-year medical oncology residents here at Texas A&M University. And I'm super excited to present the foundation of my residency project that I recently presented at Veterinary Cancer Society. And it's enrichment tools to better understand the types of circulating nucleosomes and their genome patterns in the plasma of dogs with lymphoma. And I'm also very excited about this collaborative effort that we have and all of what's to come moving forward. And so we'll start with the basics with nucleosomes. And although they were a thing of probably college and veterinary school classes, they really are quite important little structures, and they comprise the basis of the secondary structure of DNA. These nucleosomes consist of a core of proteins called histones, 4 of each in duplicate to create an octamer core around which 146 base pairs of DNA are wrapped, kind of like a garden hose on a reel. And not only do they aid in the packaging of DNA, but they're also quite important in protecting DNA from DNA damaging agents, as well as serving as a framework for epigenetic changes and therefore regulating access of the DNA to transcription factors and ultimately phenotypic expression of genes. Various studies have demonstrated elevated nucleosome levels in several disease states, including sepsis, severe burns, immune-mediated diseases, and cancer. And recently, my colleagues published a paper demonstrating the efficacy of the H3.1 ELISA to target these nucleosome levels in cancer patients. And in canine lymphoma patients in particular, there was actually a 7-fold increase in the median nucleosome concentration compared to our normal healthy cohort. The unique thing about these nucleosomes from cancer cells is that for some reason they lose the linker DNA, which is a short stretch of DNA that connects the two nucleosome cores. And it's even more important because it's associated with another histone protein called H1. And so utilizing this information, the H1 nuq capture was developed. And our goal was to utilize this capture methodology to isolate and sequence cancer-derived nucleosomes from dogs diagnosed with lymphoma, with the hopes of gaining insight into potentially diagnostic, prognostic, or maybe even targetable variations. We started with treatment-naive canine patients diagnosed with multicentric lymphoma. They were fasted for at least 6 hours prior to blood draw, and then the whole blood was centrifuged and the plasma was removed and frozen at -80 degrees Celsius prior to batch analysis. Next, nuq capture was performed. And as you can see on this slide, there's both a mixture of the long DNA within the plasma as well as short DNA or the cancer-derived DNA. Magnetic beads armed with an anti-H1 antibody were then added and allowed to incubate within the plasma, allowing the long H1 containing normal healthy nucleosome DNA to bind. A magnetic field was then applied, allowing for enrichment and consolidation of the short cancer-derived nucleosomes. Lastly, we performed paired end Illumina sequencing on both the DNA isolated on magnetic beads, as well as DNA within the supernatant or those short cancer-associated DNA nucleosomes. And this was done to an average sequencing depth of 40x. The FASTQ files were then mapped to the CanFAM4 reference genome using the BWA MEM2 algorithm to generate BAM files, and then variant discovery was performed using haplotype color and UTEC2. The lymphoma samples that we chose for enrichment and sequencing were those that had the highest nucleosome concentrations out of our lymphoma cohort. As you can see, 2,500 or more nanograms per mil of nucleosomes compared to our 24.4 nanograms per mil of nucleosomes in Chester, our normal healthy control patient. And Chester's values were quite similar to the median nucleosome concentration of the healthy cohort of dogs, which was 31.1 nanograms per mil. And in all the graphs on this slide, we have insert length on the x-axis going up to 1,000 base pairs on the top 2 graphs and to 200 base pairs on the bottom 2 graphs. And then on the y-axis, we have read pair count. The top two graphs are that of patient 10016, and on the left, this demonstrates the immunoprecipitate lengths, what we pulled out and were on the beads. And as you can see, these correspond with mono, di, and trinucleotide lengths. Whereas we can see on just our supernatant graph, we see a peak at about 147 base pairs, and that corresponds exactly to where we see the peak in our canine lymphoma patient 60010. And as you can see, there is not a peak at this location within the supernatant from Chester, our healthy normal control. Sequencing analysis of 60010's short or cancer-derived nucleosomes is shown here in the copy number variations. And in this top row, you can see what a normal baseline for a diploid genome would look like, whereas any deviations from baseline would indicate either a gain or a loss of a chromosome. And that's exactly what we see at chromosome 13, where the height above baseline is indicative of a gain, whereas the deviation within chromosome 14 below baseline is indicative of a loss or deletion. There's also a possible gain of the Q arm of chromosome 31 as well. From our sequencing analyses, there were a large number of SNPs identified, but none of them believed to be a disease associated. And so what can we gain from all of this? First, it's that we have confirmed that canine lymphoma patients have these shorter nucleosomes that can be enriched with Nu-Q capture, as seen in their human counterparts. And from sequencing these shorter nucleosomes, we've identified copy number variations, but have yet to identify disease-associated SNPs. However, not to fret, this is just our pilot study, and one of our hypotheses is that we just weren't able to get enough enrichment of our cancer-derived nucleosomes in order to really gain the best sensitivity for the SNPs that might be present. And so our hope is that as we optimize our enrichment tools, that we'll better be able to identify these SNPs and copy number variants to progress forward. And then once we have that, our goal is to extend this to other cancer types as well, starting with canine hemangiosarcoma, which actually had the highest median nucleosome concentration of our canine cancer cohorts. And I thank you all very much again for being here and again for allowing me to participate in this great collaborative effort. I hope to meet you all soon. Take care.